Thanks all for coming. Very nice to be here. So, primary question for what we're talking about today is, in the face of the challenge that cancer poses, can our cells do more? I was really tempted to have my next slide just saying no and leave it at that, but it would have been a lie. And before we can really address this question, what we really need to think about is, what does the immune system protect us against? The immune system is probably the most powerful anti-cancer mechanism in all of evolution. And, everyone has mutations, that means like errors that happen in their cells all the time. And the vast majority of us, thankfully, never know. And the reason that we don't know is because the immune system detects those errors and kills those cells whilst they're at the single cell stage, so before they can ever cause a problem. Now, so what does the immune system do? It protects us against pathogens, so bacteria and viruses, maybe. It protects us against harmful surfaces in the environment. So that could be if you have eaten something harmful, it could be harmful things that you get on your skin. The immune system is evolved in protecting the body from these factors and launching an inflammatory or a healing response in response to that. It also protects us against disease-causing changes in the body, and that is essentially what cancer is. Now, what is a T cell? This is a T cell. This is an electron microscope picture of a T cell that I took during my PhD. It is a gamma delta T cell, just for the sake of bias. I'm using it just to simply ground us in, what is it that we're talking about. But let's get a little bit more detail. So T cells, they make around 20 to 40% of our white blood cells. So the white blood cells in the body are the cells that fight infection. And they do two really, really important things. So they recognize what's called non-self. Now, that's a real problem if you're having a blood transfusion, for instance, and you get mismatched blood, you will get an immune response against non-self. It's also an issue for patients who have a transplant, because you can get transplant rejection because the transplant didn't necessarily come from you, which is why patients who have transplant need immunosuppression to help deal with that, and why we have to ideally match up the transplant as close as possible to the recipient. Now, in order to get a T cell going, in order to turn it on, you need two signals. They are very, very powerful things. So you don't want to just have them going when you poke them lightly. Then you would end up with diseases such as autoimmune diseases, where the immune system attacks the body and causes harm, as opposed to acting in its nicely orchestrated and regulated manner. So the activation of T cells is, as you can imagine, an entire lecture in its own right, and is quite complicated, but for the sake of argument, you can say that they need two main signals. So we will call those signal 1 and signal 2. And that is a kind of nomenclature that is used in immunology in general. Now, T cells are enormously diverse. So in an adult human, the T cell repertoire will recognize, as it says here, so about 10 to 100 million different things. Now, if you think about how much that is, if you take every word in every dictionary, in every known language in the world, you've got about 7 T cells per word. So it's a lot, okay? And that is in a tremendous amount of diversity, and that's really powerful because it allows you to protect yourself against all of the different bugs and all of the different problems that might happen, all of the different cancer mutations and so on and so on and so on. But it's also potentially a problem and something that we've had to overcome when we are designing new cancer therapies. So the other bit of the immune system that I'm going to talk about today are antibodies. So antibodies are not cells, they're proteins. So they're much smaller, they're not alive, you can make them in a lab, and they are also highly variable with an enormous amount of diversity, similar to the sort of diverse levels of diversity that you get with T cells. Now, some bits of the antibodies are variable, and some bits of them are constant, and variable. So this is an antibody called IgG. There are other sorts, but this is the variable bit, and this bit is typically always the same. So the variable bit, as it sounds, is the bit that has the diversity. The constant region is the bit that stays the same. That means that irrespective of what the antibody recognizes, it can dock onto a receptor, so that's like a socket, on an immune cell and activate that immune cell when it is bound to its target of choice. And antibodies circulate in the blood, they exist in our tissues, and they stick to bacteria and viruses and so on. And this is what vaccination gives you. Vaccination typically gives you an antibody response against the bug that you're being vaccinated against. So you've got antibodies floating around in your body. That means if you ever get exposed to that bug, the antibodies stick to it, they kill it before it blossoms and becomes a problem. Now, I'm mentioning these because we use bits of these in immunotherapy. So, you've got all this amazing machinery that protects you against cancer. So why do people get cancer? We're sitting here, we're going, oh, this amazingly powerful shield, and yet there's still a problem. And this is because cancer has evolved to evade the immune system. The immune system is possibly one of the strongest evolutionary pressures that is put on anything, a kind of Darwinian process. But cancer mutates. You can get new changes in every successive generation of cells within a tumor, and that means that you can potentially bypass some of these protective immune factors, and then the cancer reaches a certain size, and then it becomes harder for blood vessels to penetrate, harder for immune cells to get in, and you reach a point which is typically called immune escape, where the tumor has formed and then it starts to grow and then it becomes more difficult to treat. And this is a real problem with what we call solid cancers. So solid cancers are things that form lumps, as opposed to leukemia, which is a blood cancer which occurs in the blood. The best place for a lump-forming cancer is in the bin under the operating table, because really you want it to be chopped out. Okay? The problem is that in doing that, often tiny little bits break off that may have already spread or something like that in a way that you cannot see. And this is where things like immunotherapy come in to really try and mop up those last surviving vestiges of the cancer before they can cause more problems. So the question is, can our cells do more? Yes, they can. So what we want to do, we want prizes for working out saying which film it's from. Come on, it's bad film from the 90s. It's from Transformers. Can we train a cell from this thing on the left that's all bashed up, a little bit old, to this thing on the right that's nice and shiny and that essentially turns into a robot? Maybe not the robot. It is Bumblebee. And the question is, so how do we do that? So, and what can we do? There's a lot of metaphors in this, I'm sorry, okay? We're moving on to Lego. So we can make our cells do a whole variety of different things, and the biological machinery for doing this is available to us, and it is essentially like biological Lego. So we can give the cell steering. We can make them go where we want them to by making them home to particular signals that the body produces. We can give them cargo, so we can make them have an anti-cancer cargo. We can make them have a cargo that combats autoimmune disease. We can make that cargo stay on the surface of the cell, a little bit like giving a cell a sword, or you can have it so that it shoots out from the cell, giving a cell a bow and arrow. You can change the chassis, so you can pick what sort of cell you want to use, okay? So you can use T cells, but there are different types of T cell, and different types of T cell have different properties, and I'll tell you about that a little bit in a second. You can change what we might call the engine, so you can engineer the cells to be longer lived, so that they produce their own fuel, so that they can survive better in a hostile environment, where there aren't very many nutrients. You can also change how they might see the cancer. So how do they actually do that detection? And do they need one signal to do that or two signals to do that to change their specificity? I'm going to talk about some of those different things and how we've done them as I go through the talk. So this is kind of how to engineer a T cell, 7 easy steps. This is what we do in our lab. So you start off with a protein sequence. So the machinery in cells is comprised of proteins. Proteins are groups of what are called amino acids, and they come in a particular sequence, they have a particular structure, and they form what are essentially biological molecules. Those molecules interact with each other and then elicit a response in the cell. Now, those molecules are encoded by our genetics and by DNA. And we know that for every amino acid in here, there are these little groups of 3 bits of sort of DNA bases called codons, and they correspond, so they encode for the proteins. So it's like a blueprint for the body. We can work out what the DNA sequence might be for a protein that we want to make, because all of that has been done. The Human Genome Project has been around for 20 odd years. We know what the DNA sequences are to get what we want. And now I can go online and buy it. So I literally put in the DNA sequence that I want, I hit send, it goes off to San Francisco, and a guy in San Francisco prints it for me and ships it back. Which means that I can buy genes that I have designed myself. So I can take a bit of one protein and a bit of another protein and smush them together like Lego and get a new protein, which means I can really start doing some proper bioengineering, which is where the fun bit starts. Okay? So you get your little vial that's got your DNA in, and it arrives in the post. It literally does just arrive in the post. And I stick that in a virus. Because viruses, amazing little things, they have evolved to take their DNA and stick it into our cells to make more viruses. Now, obviously, we don't want that to happen. So we take out the viral DNA and put the new bit of DNA that we want in instead. Which means that, say, for instance, I was making a cell go green. For that, I'd use a protein called green fluorescent protein. It does exactly what it says on the tin. And I'd put that in my cell, leave it a few days, and my cell will glow green. And this works really, really well. I've got some examples of that later on, but it works really, really well. And actually, we use this green fluorescent protein often as a reporter signal to make us confident that our virus has actually gone in. So you'll have green fluorescent protein, and then your gene of interest, whatever that is, whatever you've designed in the lab. And if the cell is green, the chances are it's got the gene of interest as well. So then you can move forward. It's all well and good doing all this engineering, but then really you need to start testing it. And I really, really did try to keep fighting metaphors out of this talk, and then I failed completely because ultimately it's cells fighting cancer, so sorry. But you do then need to do what's called functional testing. So you take your engineered immune cells and you put them with some cancer cells, and you basically say, do they kill more? Do they do what they're meant to do? And that's, I'm going to show you some of the engineering designs that we use to accomplish that. So I'm going to go through these 3 examples. So chimeric antigen receptors are something that is probably the type of immune engineering that you are most likely to have heard of. So CAR T cells are something that has received quite a lot of press and has really revolutionized the treatment of leukemia. It has had substantial impact in some solid cancers, but not very many because of some of the challenges that we have in targeting solid tumours, and I'll come on to that in a bit. But it really is the prototypic cellular engineering strategy, and it's been around for a good 10 or 15 years now. There are some issues with CAR T cells, specificity being one of them. So I'm going to bring in gamma delta T cells soon and talk a little bit about chimeric co-stimulatory receptors, which potentially are safer and are more specific to cancer, which is important because it allows you to potentially target more things. And then finally, I'm going to talk about antibody-engineered killer cells. So both of these, all of the benefit is stuck to the cell. Whereas with these, the benefit is ranged. So suddenly you will unlock an enormous number of additional effectors because the cells start shooting stuff out rather than having to do all of the heavy lifting themselves. So firstly, why do we need chimeric antigen receptors? We've got this really powerful immune system. It's amazingly diverse. Well, this is the problem. It is so diverse that the number of T cells in your body that actually recognize your cancer may be incredibly small. So this is immunity in however many different languages. And, forgive me if some of the translations are off, by the way. It was ChatGPT that gave it to me, so it's ChatGPT's fault, not me. But what we really need to get to is this. Everything needs to be focused in one direction, at the cancer. And, this has been startlingly successful. But how do we do it? So you've got your T cell. So for the sake of argument, this is a normal T cell, an standard alpha-beta normal T cell, like kind of vanilla. And what you do is you take the variable bit from an antibody, because an antibody is a protein, which means you can buy the bit of it and you can buy the piece of DNA. You don't have to buy a whole cell. You take that bit and you turn it into what's called a single chain variable fragment. So if we look here, the VH and the VL bit are actually on separate chains. And that's no use to you because then you need two proteins. Whereas if you do this, you can link them together and make them into one protein, which is much, much easier to make. Okay? Then what you do is you stick it in a line, in a designed protein, with two extra signals. Now, I said to you earlier that T cells need signal 1 and signal 2. So what you do is you get the bit of protein that provides signal 1, protein that provides signal 2, and you stick them in a line in series, in essence, if you think about it in terms of electronics. And then you put the targeting bit on the end. And essentially, this is like a logic gate. So there's a bit of physics and electronics in this talk. I'm very sorry for bringing you to a biology talk and talking about electronics. But essentially, if you think, a T cell needs two signals. So it's like an AND gate. So you need A plus B. If you have A plus B, you get Q. Now, in a standard unengineered T cell, A and B are provided by different things. And that's a safety switch. With a CAR T cell, A plus B are all provided by the same thing. So you activate the CAR and you get signaling from signal 1, which typically comes from a molecule called CD3 zeta, and signal 2, which comes from a co-stimulatory molecule, which various different versions of co-stimulatory molecules exist, and they each make the cells do something slightly different, but realistically, you need both in order to get the T cells going. So what happens is the antigen interacts with what's called the scFv, so that's the single chain variable fragment, the bit that you got from the antibody. So the antigen is whatever you have picked on the surface of your cancer cell to target. So let's say it's blue, for the sake of argument. So scFv interacts with the blue bit. The CARs then cluster on the cell surface. They all kind of come together because they come together at the point of the contact with the target. The signaling domains, which are the bits of the machinery that you've put in that activate, all come together. They signal, and then you get T cell activation, and the tumor cell dies because the T cell releases cytokines, it releases killing chemicals called perforin and granzyme, and you get more T cells. So the T cells start to proliferate as well. So you get more CAR T cells in response. So you're actually building up your army as a result of them being in battle, essentially. So how do we do this? Because this all sounds very, very advanced. So what you do is you get your patient and you harvest the white blood cells from the patient. So they essentially, it's a bit like doing a blood donation. So they go and they sit and they get connected up to a machine and you gather their white blood cells. And that's called leukapheresis, leuka for white. You get the T cells from that blood that you've taken from the patient and you stimulate them with some antibodies and some chemicals. You expand them out and then you use a virus to put the gene for the CAR into the T cell in the same way as you could put the gene to make them go green into the T cell. You let them expand so that you've got enough, and then they go back into a bag, and it just looks like a kind of bag of beige fluid, essentially. And then you make some space in the patient's immune system by giving the patient some chemotherapy, and then you infuse the CAR T cells back into the patient, and then they go and attack the cancer. Now, this manufacturing process takes about 3 to 4 weeks, which is a major issue with CAR T cells, because during that time, your patient might get more sick. Because if they've got aggressive cancer, 3 to 4 weeks is a long time. And this is one of the things that we're really trying to work at solving by using other people's T cells, not the patient's T cells, okay? And I'll talk to you a little bit about that as we come on to gamma delta T cells. Having said that, it has been monumentally successful. This is Emily Whitehead. Emily Whitehead, in 2012, had horrible leukemia for which she received CAR T cells. This is Emily Whitehead last year. So she's one of the first children to receive CAR T cells against that particular type of leukemia. She's clearly doing fine. It cured her leukemia, and prior to the advent of CAR T, she would not have been able to be cured and she'd have died. And this is a story that is being repeated now all over the world as more and more people are receiving CAR T therapy. So it has been an enormous success for this type of leukemia. There's a problem though. So I said earlier on about targeting a marker, let's call it blue, okay? So you engineer your CAR T cells and you say, right, I want you to kill everything that's blue, but only the cancer, so the bit with the red box around it, that's your cancer cells, everything else is healthy. But you've told them you're going to kill everything that's blue. That's a bit of a problem really, isn't it? Because you've got all these other potential healthy cells that are also blue because they may also express this particular target, because the targets are often not that specific. And then you end up with what's called on-target, off-tumor toxicity, which is again exactly what it says on the tin. Sometimes that's tolerable. So with the commonest type of CAR T cell, it removes another type of immune cell, which you can kind of get by without. It kills what's called B cells. It's not ideal, but you can survive without your B cells, and it's probably much better than having leukemia, so on balance, you do it. But on-target off-tumor toxicity in other contexts has been fatal and has completely halted the development of immunotherapy programs, quite understandably, for safety reasons. But it does really limit what you can target, because essentially people spend billions and billions of pounds, dollars, pick your currency, finding the exact shade of blue that they're going to be able to target to get an increasingly more specific antigen. And that is really quite challenging, because you can't sample the entire body to find out if some really important cell in the little toenail isn't also blue. So you actually end up with always taking a risk whenever you do this kind of immunotherapy. wouldn't it be good if you could add a mask over this kind of colour map and say, well, have you got something that could work out if it's blue on cancer or blue on healthy? Because then this wouldn't be a problem. You'd be able to, you crack open a whole range of different targets because you'd be able to determine whether they were on the cancer cell or on the healthy cell. And that's something that we've been able to do. Because if you can do that, you can get this type of specificity, and this is what you want, because this isn't as toxic. So this really is just like electronics but squishy, okay? So your alpha beta T cells that we've talked about before, this is the conventional common T cell, they need at least 2 signals to activate. They need a T cell receptor signal, which is the really diverse bit, and they need some co-stimulation, that's the signal 2. You have to bypass that with a CAR because of that diversity, which means you end up with this essentially single switch. It's like a light switch. It's either on or it's off. And if it's on, it's killing, and then you end up with on-target, off-tumor toxicity. Gamma delta T cells, which is my one true love, and what I work on, work differently from that. The gamma delta T cell receptor is completely different from the alpha beta T cell receptor, okay? So gamma delta T cells are relatively rare. They comprise around 1 in 50 of your circulating T cells, but you can expand them. The important thing is the gamma delta T cell receptor is essentially a stress sensor. So it has a lot of diversity, but it doesn't recognize specific single things in the way that alpha beta T cell receptors do. It recognizes, is this cell stressed or cancerous, or is it not? Which is really useful because you can use that to give you your signal 1. So you don't need signal 1 in your CAR because it's already there. It's redundant. So you don't need to replace the T cell receptor. Therefore, you can separate out the 2 signals and focus on having an engineered signal 2, so engineered co-stimulation only. And that means that you can differentiate between antigen on healthy, because there's no T cell receptor signal coming from healthy, and antigen on tumor, because there is a T cell receptor signal coming from the tumor. So it looks like this. So standard CAR alpha betas, any antigen positive cell, you get both signals, you get full activation, kills the target. CCR, so chimeric co-stimulatory gamma delta T cells, in the presence of an antigen positive tumor, this stress marker is in the closed formation, and that will deliver a T cell receptor signal, so signal 1, and the chimeric co-stimulatory receptor delivers signal 2, and you still get full activation. But importantly, if you've got an antigen-positive healthy cell, the stress marker isn't activated, you don't get that TCR signal, and you get incomplete activation, which means you do not get cytotoxicity. And just to prove that it works, I've included very little actual data in this, but here is some, just to prove we do experiments occasionally and don't just talk. So, what we did here is we got some special beads, and we coated them in antibodies, like the antibodies I've talked about before, that stimulate either the T cell receptor or the CAR, or both. So you can separate out those signals like you're doing your electronics, like you're doing your logic gate. So with unmodified cells, if you stimulate the CAR, as you can see, none of the cells go up into the activated portion because there isn't a CAR there to stimulate. So that's just your kind of baseline. If you've got conventional CAR T cells and you stimulate the CAR, I'm sorry, the dots don't project that well, but as you can see, you've got basically the whole body of the cells that express the CAR, they all shift up into this bit here, which means that they're activated. If you've got CCR transduced gamma delta T cells and you provide both signals, they essentially do the same. So they move up exactly where they're meant to go and they activate. But if you only stimulate the CCR and you don't stimulate the CD3 or the gamma delta T cell receptor, they nicely sit down here without that activation, without causing any killing signals. And this is seen as well in terms of killing of disease. So on the left here, in this graph, we have CCR engineered gamma delta T cells. They're all targeting the same molecule. Okay? It's called, incidentally, it's a molecule called CD33. That doesn't matter for the sake of the graph. Importantly, monocytes. Now these are healthy cells. Everybody has these in their blood, unless you're immunodeficient, and they express CD33. All of the other cell lines on there are leukemia cell lines. And as you can see, killing of the monocytes is really, really low down here, whereas the leukemia cell lines are killed really quite nicely to varying degrees depending on which leukemia cell you have. If we look at killing of monocytes by standard CAR T cells, it's up here. So you wouldn't have any left. And this would be really toxic if you had it in a standard CAR format. But if you have it in a CCR format, you can use that essentially biologic and gate system to avoid that type of toxicity. Now, that's all well and good, but we're still stuck to the surface of the cell. So we need to break away from that, because if you're stuck to the surface of the cell, you are limited in terms of what you can hit, because you rely entirely on the engineered cell to do the heavy lifting. So this is the Battle of Agincourt, 1415. Okay? Has anyone read Henry V? School? No? Someone might have done. So Henry V, Shakespearean play, celebrated, amongst other things, the Battle of Agincourt. This is where a small force of English archers won a battle against an enormous force of armoured French knights. So you've got the little guys sitting there with their bows and arrows, and you've got the princes of France on the other side of the battlefield in all of their really expensive heavy armour, and they couldn't stand up against the English archers using a longbow. And it was a pivotal point in history that showed that the longbow was the state-of-the-art weapon at the time. But really, the point is, it's about bringing something ranged to the battle against cancer. So, I've already talked a little bit about antibodies, and the antibodies mark targets for killing by the rest of the immune system. And they do that by having the variable bit sticking to the antigen, in this case it's on a cancer cell, and then the constant bit engages an antibody receptor on any number of different types of cells. So that could be a gamma delta T cell, they're very good at it. It could be a cell called an NK cell, that stands for natural killer cell, or it could be a cell called a macrophage, which is a cell that kind of goes around and eats it eats bacteria and viruses and broken up bits of cancer cells. So wouldn't it be good if we could make cells shoot out antibody and then use the antibody that they shoot out to do killing? And this is what we've done. So I developed this a couple of years ago, and at the moment we're looking at different ways that we could start applying this in clinic. So what we did was we took some gamma delta T cells, we engineered them to secrete the antibody, and what happens is they pump it out and it coats the tumour cells, and then not only can the gamma delta T cells then stick onto the antibody and attack the tumour and kill the tumour, but anything else that can see an antibody within the tumour microenvironment can do the same. So essentially you're marking the targets to be killed and then allowing whatever can kill them to kill them, rather than relying entirely on your engineered cell to do the heavy lifting. And this really does allow you to escape this need for cell-cell contact, which is one of the challenges, because ultimately, In a CAR T product, so when you get your cells back from the CAR T factory, only around 20 to 60% of them will actually have the CAR. So the other ones in there are just kind of along for the ride. They're not really doing a huge amount. And also, they're typically made from the patient's own blood, which takes that 3 to 4 weeks, which we've already talked about. And that really limits the level of benefit you can get from a CAR T product. Whereas with an antibody secreting gamma delta, once you've reached a certain threshold of antibody production, all of the immune bystanders start coming in. So you kind of end up with potentially with this kind of exponential, or just a huge increase in the recruitment of immune cells to help attack the tumor, which potentially allows you to harness things that are already present within the tumor microenvironment. And importantly, because gamma delta T cells have a different type of T cell receptor, they do not do non-self. They're not bothered if they come from someone else, which means that you can take gamma delta T cells from a healthy person and use them to treat a patient. And that's really important because it means you can make them ahead of time. You can make a whole bank of these things, and it means that the time from deciding to give a patient treatment to actually giving them their treatment, it isn't 3 or 4 weeks, it's about 1 week. So that's, it really is potentially a more agile way of delivering cell therapy, and that's something that we're trying to get funded at the moment to really get people to be able to receive benefit from this so we can start testing it. Now, I've talked a little about engaging the bystanders, and really, I've talked about this, but what bystanders are there? So this is what's called a UMAP. A UMAP is a way of showing data where there are an enormous number of different parameters in 2 dimensions. Okay, so if you imagine a 3D picture would be just a cube or something like that, well, you can't do a 10,000D picture. Our brains don't process that. So what you do is use a mathematical algorithm to squash that down into 2 dimensions. And what it means is that things that are similar go near each other on the map. And what we can do here, so these cells are cells from a bone cancer. This is a bone cancer called osteosarcoma, which affects teenagers. And we can work out what the different cells in there are. So there's all of these different ones on the right-hand side, but the ones I want you to focus on are the T cells and the NK cells and the macrophages, because both of these, especially the NK cells, which sit around here, and the macrophages, these are cells that can engage antibody-coated target. And what we've done is we've built this computer model to show, with all of the parameters that we know from our engineered gamma delta T cells, so how much antibody they produce, how sticky that antibody is, so how well it sticks to the target, how well it sticks to its receptor, and so on, which is all data that you can find, based on all of the experiments that have been done over the years. We compile all of that together to make this, which is a predictor for where the antibody-based killing could happen within a tumor. So basically, the hotter the picture, the more potential killing power you have within the tumor, which means you can take a sample from a tumor, look at it and say, okay, if we pumped this much antibody in here with our engineered gamma delta T cells, what could we recruit? And this is showing, as we expected, that you can recruit NK cells, which we know are good at killing antibody-coated targets, and macrophages, which are good at killing antibody-coated targets. Now, in order to do that, you've got to be able to get the cells to the right place. And this is just showing that they do go there. So this is, remember I said that we mark cells and we make them green? So this is cells that we have made green. So this is a mouse leg, because trigger warning, we do work on animals. So this is a mouse leg, and this mouse has had a bit of cancer injected into its bone just here. And this one just has the tumor, so there's no real signal. You get a little bit of extra kind of green just as background. This mouse doesn't have any cancer in this leg, so it has only had the green labeled gamma delta T cells. This mouse has got cancer there, and look at where all the green signal is. So the gamma delta T cells we know will home directly to where the cancer is in these animals. So, we've got to compare this against other modalities, and we do this. This is another bit of just fun bit of bioengineering. We use something called luciferase. So this is a firefly. Fireflies glow at night, glowworm. They use a chemical called, an enzyme called luciferase, which breaks down a chemical called luciferin, and that makes them glow. So we can make tumor cells glow, and you can detect that glowing using a special camera. So we injected some tumor cells into the legs of these mice, and with that special camera, you can detect the tumor, and you get these little heat maps. This is called bioluminescence. This is one of the ways that we track tumor growth. And using that, we can then start comparing treatments. So what we did here was we looked at how long it took for the tumor size to double in these mice, and whether the tumor was going to grow or not. And we took mice with again, we gave them osteosarcoma, which is the same kind of bone tumor that we were talking about before. And the mice that received no treatment, as you can see, what we call event-free survival, so how long did they last without the tumor doubling? They got out to about 60 days before all of the mice had tumor doubling. If we gave them antibody, so just straight antibody targeting the tumor, which is actually used clinically at the moment, it really didn't give them any benefit at all. If we gave them CAR T cells, and these are conventional CAR T cells using the current kind of standard class design, it essentially did nothing. Whereas when we gave them our engineered gamma delta T cells that secrete the antibody, actually only one mouse ended up having tumor doubling at all. The rest of the mice showed essentially no tumor growth and were quite happy and healthy at the end of the experiment. So we were quite pleased that we'd managed to really show benefit above and beyond what was already being done with the kind of standard competitor immunotherapies. We've done all of this, but really, we are at the start of a journey here. So there are so many questions that are yet to be answered. So, which cells are we going to use? We're going to use alpha beta T cells, we're going to use gamma delta T cells, we're going to use NK cells. What is it that we're going to use as our chassis? Who are we going to get them from? Are we going to get them from patients? Are we going to get them from healthy donors? What are the risk benefits of that? What do we need to do in order to make those things work? What diseases are we going to treat? People are using CAR T cells to treat lupus now, which is an autoimmune disease, not just cancer. How are we going to engineer them? Are we going to do viral engineering, or are we going to move to other non-viral techniques? You can actually engineer cells by giving them electric shocks, so you could potentially do that. Do we need to engineer them outside the body? Could you actually give someone something so that their cells changed inside their body? As you inject them with a virus, and that sorts their cells out, it engineers their cells in their own blood. Are you going to get the cells to the disease site? Really important in solid cancers, especially making sure that they stay healthy along the way. And when they get to the tumor, tumors are really hungry. They take up all the oxygen, they take up all the glucose, they spit out a load of acid and make the immune system fall over. So you're going to have to make a way of ensuring that your immune cells can survive that. And then what do they do when they get there in terms of specificity and engaging bystanders? And that's what we're working on at the moment. We really hope to be able to take this forward. And yeah, it's been a fun journey. So just like to thank a whole load of people, all of the guys in my group, and our collaborators, and of course our funders, and Fran for inviting me to come and bend your ears for an hour or so. Yeah, thank you very much.